Good afternoon. This is Dr. Dan Guerra coming to you from VerEvMed Studios in the early days of 2019. In fact, it is the 23rd of January, the second installment of my free lectures that I'm doing for this year. I've been putting out on Facebook that what we're going to be doing this year is moving to a podcast format. That doesn't preclude these video lectures. We'll still do these, but the podcast will be an integrated online interactive discussion of published literature in, again, biomedical journals and affiliated natural sciences. So we're going to do this podcast, and it means things will change format wise. I'm not sure what day of the week we're going to launch it, but it will be something convenient, a convenient time of day, and we're going to get this thing rolling. So we're still VerEvMed. We're doing the same thing we did before, but we're adding another plank to the system of the VerEvMed studio archive. So I also mentioned in many of my previous posts on my Facebook page, which is shown down here, that what I'm going to do is start off this lecture series doing my normal lectures, that is pre-taping them using YouTube as the platform for you to see them. And then hopefully somewhere in between the beginning and the end, we're going to switch to some of the discussions at podcasts. We're going to blend the two. So you'll see some video and you'll also start hearing the podcast. And hopefully we'll be able to put on the podcast, maybe often or not, some video. And certainly when we want to emphasize things, we will have some slides that maybe will either be in the show notes or maybe will be also some kind of semi-live preparatory system with the podcast. So let's get started, shall we? All right, so okay, I'm Dr. Daniel J. Guerra, and I am the co-founder and the chief scientific officer and honcho of VerEvMed that I co-own with a collaborator and a pharmacist. And what we do is we look at the primary scientific literature and we answer questions for clients. The clients can be physicians, they can be technicians, they can be researchers, they can be entire institutions like hospitals or nursing staff. We also consult with departments at various universities and, of course, with individuals. With individuals, it includes all the people I mentioned, but also lawyers, and also, very importantly, regular folks who want to know more about what the primary scientific literature is saying about nutrition and about all aspects of biomedicine. And that includes psychiatry and neuropsychiatry, and maybe even a little bit of psychology. I'm a biochemist, a lipid biochemist, but what I try to do is look at the primary published literature in the journals that are out there that are all refereed, so it's well vetted, look at the evidence in those journals, and then discuss it at length with our clients because they have questions about, let's say, a new therapy or a new drug, or they just want to know what's going on, for example, in oncolytics or in the bioenergetics of yeast. It could be anything. It could have something to do with trypanosomes. It could have something to do with cardiovascular disease or cancer, obesity, or diabetes. And what I'll do is I'll go into this primary literature, I'll find publications that are emblematic of that particular subject matter, and I will pull out what the evidence says in those papers, completely dissect them, and then verify that evidence. This is another big thing we do. That's why we're called VerEvMed. We verify the evidence against the backdrop of all the other evidences out there in a given discipline, so that you're not just getting brand new material, but that is what we want to look at, the stuff that's published more recently. But we're going to go back and we're going to look at the archives all the time because we want to know what the corpus of the literature says about a given topic. That way you get the full breadth of an understanding of a biomedical problem or a scientific research issue or maybe a legal issue between two clients discussing something that's related to sciences. Okay? So again, I'm going to spend some time talking about VerEvMed, just that, full stop. But right now, I want to get into this topic for today. It's going to be a multiple-part, multi-segment VerEvMed lectures. So there's going to be mini lectures. I'm going to put them all together eventually, and they'll all be posted on my Facebook as YouTubes. Let's get started. So today we're going to talk about bioenergetics. And bioenergetics has to do with the cell's ability to synthesize compounds that have a high energy potential. Primarily, we're talking about ATP, adenosine triphosphate. But we're going to find out that there are other high energy intermediates. Often they are phosphointermediates that do provide enough energy to drive chemical reactions. And in the cell, they're biochemical reactions. And if they're embedded in pathways and their entire systems, such as the oxidation of glucose to pyruvic acid, or the oxidation of a fatty acid to acetyl-CoA, or the synthesis of DNA from purine and pyrimidine nucleotides, or the synthesis of RNA from DNA template. You get the idea. So today, bioenergetics is going to be a broad, general sweep of what we mean by it. As a biochemist, I taught biochemistry for about 3 decades at university, mostly at the graduate level. And so what I'm going to do is go into my repertoire of being just a professor of biochemistry and let you know what we mean by bioenergetics. Then what was going to progress with multiple lectures from here on is eventually we're going to get to a couple of interesting papers that were recently published this year, in fact, just this week in major journals. So there again is my email address. That's my private email address. You can send information and questions directly to me at djgphd@gmail.com. This is my Facebook for the VerEvMed. You can take a look at that. All the previous video lectures, also a lot of other information, is on that Facebook. It's kind of like a catch-all for everything that I do professionally or even semi-professionally, because I also do write in philosophy. Today's date is 23rd January. Let's get going. Ultimately, what I want to do at the end of this series of lectures is talk about lipid signaling and how it controls ATP transport. Remember, ATP is a high energy intermediate in the cell whereby biochemical work can be done in the cell and therefore in the system. And we'll discuss all about what we mean by bioenergetics and even the biochemical work as we proceed. So first of all, we're going to talk about bioenergetics. Sorry about that misspelling. Then we're going to talk about sphingolipid metabolism, because we're going to find out we need to go back and review our sphingolipid metabolic pathways, because that's going to be the key lipid involved in controlling ATP transport across the intermitochondrial membrane. And specifically, it's going to be ceramide, that particular sphingolipid. And we're going to see that there's a ceramide-mediated organelle fusion and turnover. By organelles, I mean mitochondria, endoplasmic reticulum in particular, but also components of the nuclear envelope and the plasma membrane, and even the Golgi and peroxisome. So that'll be the third segment of this broad sweeping series of lectures. Then we're going to go back and review autophagy versus apoptosis. How a cell digests itself during times of starvation is really important, and what happens after that, whether or not the cell becomes proliferative or becomes senescent, for example, key feature. And then the sort of opposing arm of degradative pathway would be programmed cell death. One particular type is apoptosis. Apoptosis is that kind of programmed cell death, which ends up with dead tissue, dead cells. And we're going to see how those two, autophagy and apoptosis, are actually kind of at opposite poles when we talk about cellular fate, as associated with disease and related to, in this particular case, the bioenergetics of the cell. Next, we're going to go back and recap Alzheimer's disease, because ultimately that's where this is all going to be leading to. One of the major biomedical concerns, of course, is late onset, age-related Alzheimer's disease, neurodegeneration, leading to a lack of cognition or a loss of cognition, and ultimately, of course, to death. Rather an unfortunate way for people to die. So we're going to eventually key in on that really important biomedical pathology, and all of this is going to lead to that, okay? And then finally, I'm going to give you my diaventimological perspectives. And if you've watched my previous lectures, you know that I've developed a new paradigm in science called dia-event ontology. And what that is basically going to do is bring together, or bring all the threads together of a given topic, and talk about how space and time are related to a discussion of anything related to science. And you'll get what I mean more when I talk about event ontological issues. Right now, just keep that in brackets because we're going to get to that later on. So let's just talk about bioenergetics, the bioenergetic background. There it is spelled correctly. All right. So there are two basic things that happen in living systems, catabolism and anabolism, where catabolism is the breakdown and anabolism is the synthesis of organic compounds. So during process of catabolism, energy is produced. Very important point there. Carbohydrates, lipid, and protein are all going to be the molecules that will go through the process of catabolism, that is the breakdown. The ultimate products of that, besides energy in the form of ATP ultimately, are going to be carbon dioxide, the fully oxidized form of the more reduced carbon found in carbohydrates, fatty acids, and amino acids, which compose protein. Water, of course, which is going to be sort of a byproduct of that oxidation process, where we're going to reduce molecular oxygen from the atmosphere ultimately to water, okay, through a several electron, one-step electron at a time reduction of oxygen to water. That's part of the process. That's in, again, the mitochondria. And of course, the production of ammonia, which is going to be involved in waste removal in the form of urea. So if you use an amino acid for bioenergetics, for energy production, and in fact, whenever you interconvert amino acids, which of course is the building blocks of proteins, you have to worry about carrying around the amino group, the NH3 group, because amino groups can be very toxic. Think about ammonium and ammonium salts, for example, that are used in cleaning solutions. They're very toxic because of what they do to cells in terms of the vagaries, the effects they have on membranes and macromolecules like DNA, RNA, lipids, and proteins. And we can talk a little bit about that too, the toxicity of ammonia. But those are the major, basically inorganic products of catabolism. In the process, the bioenergetics of the system allow you to make ATP, and NADH is nicotinamide adenine dinucleotide, and then NADPH, which is nicotinamide adenine dinucleotide phosphorylated form. And the H stands for a hydrogen atom, meaning they're both reduced. In fact, NADH is going to be reoxidized in the electron transport chain, and ultimately protons pass through the intermitochondrial membrane in the proton pumping ATPase to make the ATP. And NADPH has a more of a complicated story. It's basically the biological reductant in the cell, and in fact, removing some of the partially reduced forms of molecular oxygen, which are going to be intermediates in the chain of the pathway taking molecular oxygen, O2, all the way to water. So one of the major issues of NADPH is to eliminate those partially reduced forms because they can be quite toxic, again, to nucleic acids, proteins, membranes, etc. And that's also part of the aging process and also related to the Alzheimer's disease, as we will eventually see. Now, you need energy to carry out anabolic processes, and those are the processes where you resynthesize organic compounds. All cells are constantly breaking down certain high energy, highly reduced forms of carbon to carbon dioxide, water, and ammonia. And then ultimately, the energy produced from that system either does physical work like muscle contraction or synaptic cleft activity, which involves, of course, the processes of reasoning, or all kinds of other issues that require energy. Just movement of ions and movement of small molecules across membranes requires energy. So that's what the energy is going to be used for. But likewise, the energy is used to rebuild organic compounds. And you might say, well, why ever break them down at all? Why not just spare them? It's because macromolecular structures age, just like structures out in the world age, like a bridge or a road, and they need to be rebuilt. And the way you rebuild it is you start from scratch. You break it down to individual monomeric units and you build it back up, and that requires energy. And that's going to be in the form usually of just plain old ATP. But then NADPH is also often used as the reductive biosynthetic molecule. And we'll see that, for example, in fatty acid synthesis. Again, we're going to cover all of this in a highly truncated form because to cover this whole thing would be practically a semester in biochemistry. We're not going to do that entire thing. Anyways, in anabolism, You synthesize macromolecules, as I said. You also carry out muscle contraction. You move ions across, and then also there is some thermogenesis, right? We are organisms which control our own heat, right? And because of that, because we can control our own temperature, we don't require any external temperature. So that whole process requires energy. And so there's shivering and non-shivering thermogenesis. The non-shivering actually does involve brown fat, which you probably have heard of, and an uncoupling process which allows for the production of heat directly from translocation and utilization of fatty acids, and then in the ultimate process generating heat rather than the movement of protons and electrons through the electron transport chain. Again, we'll get through that a little bit too. The products then of anabolism, besides all that great anabolic work, is of course ADP plus PI. So we've removed the gamma phosphoryl of ATP. We've oxidized the NADH back to NAD+, that's what that means. And we've also oxidized the NADPH to NADP+. And then those get used again in catabolic processes, and they get the two dinucleotides get reduced and ATP gets resynthesized. So that's a basic background in this bioenergetics. So what do we mean specifically? We're talking about adenosine triphosphate over here on the left, and adenosine diphosphate. So adenosine diphosphate just has two phosphates. So this is adenine, the purine nucleotide. This is the ribose sugar. So this is a ribonucleotide. And these are the two phosphates, right? And so this is adenosine 5' diphosphate, because that's where the two phosphates are, the 5' carbon there of that ribose sugar. Now, when you add phosphate to that, you make ATP. You take this diphosphorylated form and you make one more phosphoanhydride bond right here. That's the gamma phosphoryl that's going to be used for most of the bioenergetic work. Sometimes we also use this second phosphoryl, this beta phosphoryl, when we're making just AMP, because we can use the energy of both of those bonds to drive work and to drive chemical synthesis. But this is basically the whole system when we talk about ATP synthesis from ADP and PI, or the breakdown of adenosine triphosphate to ADP and free PI, that's inorganic phosphate. So what kind of molecules are we talking about? Carbohydrates and lipids, those are the most common ones. And we think about carbon at various oxidation states. The oxidation state is important to consider, because the more reduced form of the molecule, such as a lipid, where all these carbon bonds are saturated, we call it saturated, just with hydrogen atoms, that's more reduced, which means you're going to get more energy out of that. So burning a fatty acid gives you more energy than an amino acid or a carbohydrate. I'm sure many of you remember that even from high school chemistry or high school biology. Now, carbohydrates can exist in various forms. They can exist as alcohols. That's an OH group here to this carbon. They can act as ketones. This is carbon double bonded to oxygen, right? Or they can be aldehydes. Okay, they can be aldehydes. They can be aldehydes, ketones, or alcohols. Those are the three most common types of carbohydrates. And they are more oxidized than lipids, so you get less energy from the carbohydrates when you break them down, when you go through catabolism. Again, what catabolism means is taking a reduced substrate and oxidizing it, such as glucose, and oxidizing it to 2-pyruvates. That's the glycolytic pathway. At the same time that occurs, you can take NADP and make NADPH. Okay? Once you make NADPH, that can be used in anabolic processes as a reducing equivalent to take some oxidized precursor and reduce it to the product that ultimately is going to be made. Now, this pathway using NADPH, okay, is more common not in glycolysis, right? Although there are some reactions other than just the oxidative pentose phosphate shunt. But the oxidative pentose phosphate shunt will take NADP and make NADPH. In other words, it will reduce that particular nucleotide diphosphate. At the same time, it would decarboxylate and oxidize glucose 6-phosphate. That's the OPP pathway. And we'll briefly take a look at that. And then the NADPH, again, for example, we want to plug something in here, can take something like acetyl-CoA and malonyl-CoA, and by pumping in electrons, produce fatty acids by chain elongation. a three-step process that's repeated iteratively. All right, now, what are some of the key reactions that we talked about in terms of just getting energy? Well, many of them are enzymatic reactions, which involve intermediates, which you see throughout the cell. So some of these are glycolytic enzymes. For example, 1,3-bisphosphoglyceric acid has two phosphates, can synthesize ATP. It's a substrate level phosphorylation, we call that, substrate directly from the substrate of the reaction, you see. This is called phosphoglycerate kinase. So one of those phosphates get lost, and you're left with just 3 PGA, but you make ATP. Okay, that's a glycolytic enzyme. Here's another glycolytic enzyme, pyruvate kinase, which takes this high energy intermediate enol compound and turning it into this carboxylic acid, pyruvic acid. Now, when that happens via the reaction of pyruvate kinase, you can also do a substrate level phosphorylation. What other kind of tricks can you do with ATP? Many of you that are in nutrition or in exercise physiology, exercise science, know that you can store ATP in the form of phosphocreatine. So that's taking ATP, that gamma phosphoryl, and adding a gamma phosphoryl to this amino acid product, creatine, and phosphorylating it so you make creatine phosphate and ADP. Now that reaction is freely reversible. That means that you can take then creatine phosphate in, for example, the muscle tissue and resynthesize ATP and use that ATP directly for muscle contraction. Okay. Another, and notice we're looking at delta G naught of each of these reactions here, at least these last two. Whenever you get a Gibbs free energy, which has to do with the enthalpy and the entropy of the reaction, which involves basic chemical thermodynamics, whenever you get a Gibbs free energy in biological systems that's positive, it means the reaction doesn't want to go favorably. You're pushing against it because what you want to do is have a negative delta G. Negative delta G means the reaction prefers to go in whatever direction the arrow is showing. So this particular reaction, glucose to glucose 6-phosphate, is slightly, right? It has a negative 4 kilocalorie per mole. So this reaction wants to proceed from glucose to glucose 6-phosphate. Without anything else happening, this enzyme can carry out this catalysis because it's favored because of the basically the thermodynamics and the bioenergetics of that reaction. We can explain that too, down to the granular molecular form when we want to. This reaction, essentially though, see, because it's positive in this direction, basically doesn't want to make phosphocreatine. It wants to go back the other way and make ATP. So this is highly favored going in the backward reaction. You can tell that because it's a positive delta G prime going from left to right. So that would mean it'd be negative, exactly negative 1.5 kilocalorie per mole if we went back the other way. All right. This gives you some idea now about the bioenergetics of just breaking down any of these organic compounds. Glucose, there's a molecular mass at 180 Daltons versus lactate, which is 90, which is then palmitic acid, 256 Daltons. Tripalmitin, which is 3 fatty acids of 16 carbons, no double bonds, a palmitic acid, bound to, as an oxygen ester, glycerol. So that's called tripalmitin, which is a triacylglycerol. Tripalmitoyl acylglycerol is what it is in long written form. There's a much more amount, there is a larger molecule, but look at the amount of energy you get from breaking it down compared to glucose. Almost 32,000 kilocalorie per mole versus only about 3,000 of glucose, right? That's about like, what? How much more is it? 10 times more than 10 times more the energy from breaking down one triacylglycerol molecule, which stores depot fat in your viscera, for example. Also lots of other places, the muscle, for example, and around endothelial cells, which is usually not a good thing because that can lead to atherosclerosis, which we can also discuss later. Even palmitic acid, you can see here, is about 3 times more potent in terms of energetics than glucose, and same and even more so of just this plain old carboxylic acid, lactic acid. So that's the kilojoules per mole or kilocalorie per mole. You can see the conversion ratios there. We can talk about kilojoule per mole, but since so many people talk about calories, us biochemists still refer to them. Okay, and those are then caloric values of each of these compounds, right? And you can see here again, 9.3 kilocalorie per gram of tripalmitoyl acylglycerol versus 3.8 kilocal per gram of glucose. You can see that you get much more from storage lipid than you do from storage glucose. So this is the preferred fuel when you can access it. Okay, there are a lot of reactions that we need to talk about. Enzymatic reactions, okay? Protein associated, usually enzymes are proteins, not always, but almost always. Sometimes they're RNA. Sometimes they're metalloprotein complexes. And sometimes they're even lipid associated protein complexes, and even glycolipid membrane protein associated complexes. But enzymes in general, if you want to just think about what the card-carrying normal enzyme is in a eukaryotic cell, one of your cells in your body, are going to be proteins, right? Which are going to be polymers of amino acids. Sorry, I'm breaking this down real simply to those of you who have had this 100 times, but I just want to make sure I'm including more and more people. Now, the mitochondria is going to be where the site of all this bioenergetics is going to occur. It's where you're going to make ATP, but also where you're going to oxidize NADH, and it's also where you're going to reduce molecular oxygen to water. So a lot of things are going on in the mitochondria. That's why sometimes they call it the powerhouse of the cell, which I don't like to use. And so first and last time I will use it in all these series of lectures. Because the mitochondria does so much more than just generate ATP. It's an important place for synthesis of a lot of the organic molecules we talk about in biochemistry. But it's also in constant communication with the nucleus, the endoplasmic reticulum, the Golgi apparatus, and what's going on in the nuclear envelope and the plasma membrane. So the mitochondria and the peroxisomes are as well, and even the autophagal lysosomes. So the mitochondria is a very important organelle, not just involved in the synthesis of ATP. But I want you to see, even when you just think about that one organelle, the mitochondrion, it itself has 4 different compartments with its own discrete suite of proteins. That means there's division of labor. So that's called compartmentalization. When certain enzymatic reactions happen only in mitochondria in total, that's a compartmentalized system, such as the electron transport chain, and oxidative phosphorylation, beta oxidation of fatty acids. 3 of those things come to mind immediately. But within the mitochondria, notice there are also specific segments of the mitochondria, or subsections, or suborganellar regions. Those include the outer membrane, the inner membrane space between the outer and the inner membrane. The inner membrane itself has really basically all the beta oxidations set up for fatty acids, and also, of course, all the electron transport chain proteins, all complexes, all 5 complexes. And then the matrix, which has to do with some of the key enzymes, such as, most importantly, the tricarboxylic acid cycle. Okay? So, and there are a lot of other reactions. The urea cycle enzymes are in there. There are reactions of nucleotide metabolism. There are reactions of heme biosynthesis. It's just a whole suite of enzymes that are in the matrix. That is what's inside both the double membrane space. It's actually inside the mitochondrion. So you can take a look at some of these enzymes. The outer membrane has monoamino oxidases, kinorine hydroxylase, a phospholipase, fatty acyl-CoA synthetases. You add coenzyme A to a fatty acid so it can be utilized and put in a biological play, biochemical play. One of the key enzymes in the electron transport chain, NADH cytochrome C reductase. That's one of the last enzymes, is terminal reductase, which is rotenone insensitive. There's also one in the inner mitochondrial membrane, which is sensitive to rotenone. Choline phosphorotransferase, making phosphatidylcholine. The intermembrane space has very important enzymes. Adenylate kinase, that's going to make some ATP. Actually, that's going to make ATP from 2-ADP, right? So you can take ADP and make ATP and AMP. That's one of the reactions of adenylate kinase. There's a nucleoside diphosphate kinase, which is very much like I just described there. Any nucleoside diphosphate can exchange the gamma phosphoryl between mono, di, and triphosphorylated nucleotides. Those can include ATP, CTP, UTP, GTP, and of course ATP itself. So all 4 of the nucleotides that you might consider, well, actually all 5, even the ribonucleotides. Intermembrane has all these key enzymes involved in the electron transport chain. There's the F1F not ATPase. That's the one that actually is the proton pumping ATPase for making ATP. The NADH dehydrogenase, so-called complex 1, electron transport chain. Beta-hydroxybutyrate dehydrogenase, key enzyme in ketogenesis. The cytochromes, all these proteins are embedded in the inner membrane. Carnitine acyl-CoA transferase, that's moving fatty acids in and out of the mitochondrium. So acyl-CoA transferase means you take a acyl-carnitine and convert it to an acyl-CoA. That acyl-CoA inside the mitochondrial matrix, for example, then would be used for beta oxidation. You have adenine nucleotide translocases. You have to move the ADP around. There's monodi and tricarboxylic acid transporters, such things as movement of citrate, malate, and also things like aspartate and glutamate, and alpha-ketoglutarate and oxaloacetic acid. Those are amino acid, alpha-keto acid cogeners. There's a glutamate aspartate transporter, which is really important for controlling redox and also for controlling bioenergetics. And of course, there's the glycerol 3-phosphate dehydrogenase, which we'll talk about, which is a key enzyme player for moving NADH by not moving the nucleotide itself, but it's reducing equivalents across the membrane into the mitochondria, being generated from glycolytic pathway. Then the matrix has all these. These are mostly TCA enzymes, tricarboxylic acid, or Krebs cycle enzymes. The pyruvate dehydrogenase, which makes acetyl-CoA, citrate synthase, which takes OAA, and acetyl-CoA makes citrate. The isocitrate dehydrogenase, the alpha-ketoglutarate dehydrogenase. These are both involved in making NADH. A connotation is just the change of the substitution of carbon, where the OH group fits on. It's setting up for the dehydrogenase reactions. Fumarase is just moving around electrons through a double bond system. Succinyl-CoA synthase, malate dehydrogenase, another NADH synthesizing enzyme. That is the last enzyme if you're thinking about the Tca cycle as a clock. It's the 11 o'clock. All the beta oxidation enzymes, which we'll talk briefly about. Glutamate dehydrogenase, which is going to be involved in utilizing amino acids for bioenergetics. Glutamate OA aminotransferases are called transamination reactions. Key, and we'll see where they fit in. The ornithine transcarbamylase, a urea cycle, carbamyl phosphate synthase 1 urea cycle, and heme synthesis. Some of those enzymes are also in the matrix. All right. Now we also have to think about the electrochemical potential of these reactions. So we talk about standard reduction potentials, and what that means is the tendency for a chemical species to be reduced. Okay, the standard reduction potential is the tendency, the availability, the possibility of a given organic molecule to be reduced. Okay, and it's measured in volts. That's why it's called an electrochemical or standard reduction potential. And it's measured in both its standard conditions, which has to do with pH. The most positive the potential is, the more likely it will be reduced. Okay, so when you have a highly positive standard oxidation reduction like 0.82, this is going to be in volts, right? Taking oxygen and reducing it to water is a very favored reaction in terms of the standard reduction potential, whereas turning acetate into acetaldehyde is very unfavorable. Okay, so it's kind of the opposite when you think about the Gibbs free energy in terms of the sign. All right. Now, many of these reactions you're looking at here are the reactions that are involved in the electron transport chain. Some of them aren't, such as these are components of digesting alcohol. So ethanol dehydrogenase and acetaldehyde dehydrogenase, those reactions also are fitted in here because they all have a standard oxidation reduction potential, which are linked to what's going on in the mitochondrium when you intoxicate the cell with ethanol, for example. And you can take a look at NAD going to NADH. That's like unfavored in terms of standard reduction potential. It wants to go the other way. That's good because it's the beginning of the electron transport chain. And you take a look at some of these cytochromes, B, C, and A. They're all getting slightly positive, which means that they're favored. Okay, they're favored. That means you're moving along during the later stage of the electron transport chain. The standard reduction potential wants you to move down that gradient from very electronegative to more positive. And the more positive, the more likely it's going to get reduced, the more you're going to be moving electrons, because moving electrons is a reduction potential. So one way to quantify whether a substance is a strong oxidizing agent or a strong reducing agent is to use that oxidation reduction potential, or we just call that the redox potential, not the oxred. That doesn't sound too good, so we call it redox. Strong reducing agents can be said to have a high electron transfer potential. Strong oxidizing agents have a low electron transfer potential, which makes sense, right? If you're strongly oxidizing, you're not going to be moving electrons away, right? It's not going to be able to transfer electrons away because you're already oxidized, right? Likewise, reducing agents have a high electron transfer potential for the same kind of reason, only in the reverse. Oxidizing and reducing agents occur usually as couples. That's what we call it the redox, with a strong reducing agent coupled to a weak oxidizing agent and vice versa. And we'll see those couples coming up here. Since these processes involve the transfer of electrons, the measurement of the resulting charge separation, because that's what you're going to get here, can be quantified in voltage measured between the couple and the standard hydrogen half cell, which is just standard procedures for biochemical laboratories. And the standard for hydrogen reduction half cell is 1 molar hydrogen and 1 atmosphere of hydrogen pressure on it. That's the standard reduction pole, right? That's the half cell that we use as the standard from which all those other reduction potentials are gathered and generated. So the redox potential of the hydrogen is set at 0 at pH 0, but for tabulations at pH 7, which is used for the hydrogen under those conditions, its redox potential is actually negative 0.421 volts. Okay? Because we talk about pH 7 because that's the pH. Remember, that's going to be the amount of free protons, right? It's going to be in the cell. So a pH of 7 is called neutrality, and most cells hover between 6 and 8, but usually between something like 6.8 to 7.4 in terms of the pH, right? Again, that's the negative logarithm of the proton ion concentration, which again, I'm sure you've learned in high school, and I know many of you know what it is. The redox potential of couples that are commonly occur in biochemistry give insight into the roles in biological energetic processes or bioenergetic processes. A negative number for the potential indicates a reducing agent, of course, whereas a strong oxidizer will have a positive redox potential. Since the obtaining of useful energy in a biological process generally involves the oxidation of a foodstuff, it follows that a large redox potential, like negative 0.32 volt for NAD, suggests a useful role in producing a reduced product like NADH. Okay? A reduced coenzyme 8, NADH, then can be reoxidized, to provide energy for a biological process. So here's some of those enzymes we talked about a few minutes ago when we were tearing apart the mitochondria. There's the nucleoside diphosphate kinase taking any deoxy NAD or ribonucleotide like NADP and turning it into an NTP. So you've added diphosphoryl to triphosphoryl, and that's going to be used in biosynthetic activity. At the same time, you're going to be taking ATP and making ADP, right? So you're going to add the phosphoryl to whatever this nucleotide is, either a ribonucleotide or a deoxyribonucleotide, that's what D stands for. So that's what nucleoside diphosphate kinase does. Whenever you see the kinase, that means it's group moving around the phosphate, inorganic phosphate. And energy is required to do this. Energy is generated in this reaction when you go back the other way. Okay, so nucleoside monophosphate kinase will take nucleoside monophosphases and turn them into diphosphates. Okay, and again using ATP as that group potential source of inorganic phosphate. Finally, another enzyme to consider is in muscle. That's why it's got the myo in front of it. Whenever you see MYO, that means it's in the muscle. You can take ATP plus AMP and make 2 ADP. Likewise, you can take, and we already talked about this reaction with DNA kinase, you can take 2 ADP and make 1 molecule of ATP from it. No energy is lost or gained here. As you can tell, it's just moving around that group, the phosphoryl group, making those phosphate hydride bonds. Okay, blowing it back up now, we think about 3 major storage forms, with protein not really a storage form, but a labile, structurally efficient cause of production of acetyl-CoA. Glycogen, of course, is a monomer of glucose. Triacylglycerol, sorry, it says triglyceride. Many biochemistry books still rename it incorrectly. It should be called triacylglycerol because you don't have 3 glycerides, you have 3 fatty acids. The acyl is the particular kind of radical bond that you get with that oxygen ester. At any rate, glycogen through a process called glycogenolysis will produce just glucose because it's a homopolymer glucose. Glucose will go after phosphorylation, moving around the glucose from glucose 1 phosphate to glucose 6 phosphate will go through glycolysis, generating pyruvate. That's an oxidation pathway. Pyruvate dehydrogenase making acetyl-CoA. Likewise, triacylglycerol will go through a lipolysis where you're passing water over those bonds, right, over those oxygen ester bonds. When that happens, you get a free fatty acid. The free fatty acid has to be esterified to coenzyme A for beta oxidation to occur, making a thioester now. And then that's all going on in the mitochondria, making acetyl-CoA. Protein is broken down by proteolytic enzymes to individual amino acids. Those individual amino acids can be deaminated directly, or they can go through oxidation reactions, ultimately yielding acetyl-CoA. So you can see that everything leads to that really important central intermediate, acetyl-CoA. So carry it out a little bit more specifically now, fine-tune it a little bit. If you think about glucose going through glycolytic pathway, pyruvate has multiple fates. Pyruvate can transaminate with another amino acid, making alanine, because pyruvic acid is the same carbon skeleton as the amino acid alanine. So if you add an amino group to it, an alpha amino group to that alpha carbon, you make alanine, alpha amino acid. You can directly reduce pyruvate and move it out of the system. This is what the muscle does when it goes through a lot of contraction, skeletal muscle. Lactic acid enters the Cori cycle, for example, when it enters the blood, goes to the liver where it's used as a source of glucose in a process called gluconeogenesis, where the synthesis of the glucose requires NADPH from the oxidation of fatty acids. You can also, one of the fates of pyruvate can be carboxylated. That's an important enzyme in the Tca cycle where you make OAA, mitochondria, or it can be oxidated decarboxylated via the pyruvate dehydrogenase. And we already looked at this, and we're going to look at it in more detail. That's the production of acetyl-CoA. Okay. Now, when you think about coenzyme A, okay, coenzyme A itself is a much larger molecule than just this little bit of 2-carbon, this acetate group, which is going to be activated by this huge molecule because of this thioester bond. High energy thioester bond is going to allow this group, this acetyl group, to move around and, for example, condense with such things as bicarbonate to make mal-CoA, or condense with more mal-CoA, release of CO2, to make acetoacetyl acyl carrier protein, or acetoacetyl-CoA, depending on what system you're looking at. So the rest of this molecule involves beta-mercaptolethamine, pantothenic acid, which is a vitamin for humans. We don't synthesize this. It actually comes from amino acid metabolism. And then the rest of it is just, look at this. It's just ADP, right? There's adenine. There's a ribose sugar. There's that phosphate group. It's a diphosphate attached to the pantothenic acid, which is attached to the mercaptolethamine, which comes basically from amino acid serine, from sulfur-containing amino acid cysteine. So that's a cysteine residue. This is pantothenic acid from the diet, and this is just plain old ADP, and that is all coenzyme A. So ultimately, I'm going to close today, the first series of lecture, getting to the Tca cycle. Here is the Krebs or Tca or citric acid cycle, showing you the actual structures. When you take biochemistry, you know you're taking real biochemistry when your professor shows you structures. If you learn the structures, especially if you come from an organic chem background, or even if you don't, biologists know all about structure-function relationships as well. You can understand all the bioenergetics in a much more simpler mode of understanding than if you try to memorize the reactions and memorize the structures. I never understood why professors suggest that students do that, or why students do that. But unfortunately, nowadays, they don't even show you structures. With the highly condensed forms of biochemistry they teach, for example, some physicians in medical school, they would never even see any of this. They would presume they saw it somewhere before. I think that's a huge mistake. At any rate, pyruvate, going through the pyruvate dehydrogenase reaction, making acetyl-CoA. It can also be carboxylated to OAA. Okay, so OAA and acetyl-CoA. OAA is oxaloacetic acid, you see here. will condense to form citric acid. That's the enzyme called citrate synthase, the first real enzyme of the Tca cycle. Citric acid then can be, we're going to move around. Notice, look at this molecule here, citric acid. All right. We're going to move this hydroxyl group around, ultimately down to this carbon. It was on the 3 carbon. We're going to put it on the 4 carbon now. 1, 2, 3, 4. Actually, it's the 5th if you count this carbonyl here. But the basal structure here, this carbon here, is going to pick up the OH group where it was on the 3rd coming from the top. It's going to now be on that carbon down there. So the whole process of the aconitase enzyme is removing water, making this double bonded cis-aconitate, which never builds up. And that double bond allows you now to redirect, okay, because of isomerization, putting the hydroxyl group now on that carbon atom. That leaves you with isocitrate. This gives you the right oxidation state of this molecule, okay, the right structure to go through the first dehydrogenase. Isocitrate dehydrogenase has enough reducing power to reduce NAD to NADH. You're going to make another intermediate here called oxalosuccinate, which will spontaneously decarboxylate. Okay, it's going to decarboxylate, going to lose that carboxyl group right there. Right? And you're going to form the first CO2. This is part of respiration, right? It's happening all the time in all eukaryotic systems. All living systems carry out respiration where they completely oxidize carbon to CO2, right, in the presence of molecular oxygen. All aerobic systems do this. Isocitrate dehydrogenase then carries out both functions. It carries out the reduction of NAD and NADH, and it carries out the decarboxylation. So it's a decarboxylating oxidoreductase is basically what this enzyme is. It's a dehydrogenase. That's what they do. Now you're left with alpha-ketoglutarate, right? So you've got rid of one of the carbons already. Remember, this is a catalytic pathway, a catabolic pathway, excuse me. Alpha-ketoglutarate now is going to go through something, a reaction called the alpha-ketoglutarate dehydrogenase, where it's going to now utilize coenzyme A. It's going to use the thioester group potential of this reaction, and it's going to drive off carbon dioxide one more time, okay? And it's going to put this coenzyme A and make a thioester onto that carbonyl there that used to have this carboxylic acid on it. So now you've made a thioester, which is a more higher energy intermediate, because now you're at a point where you've done some oxidation and the molecule is running out of reduction potential. It's running out of Gibbs free energy potential. So you're kicking back energy in the system by making a thioester and driving the reaction with loss of carbon dioxide. At the same time, because you made a thioester, you can reduce NAD to NADH. Second time you've done that, you'll see why that's important soon. Then you've got succinyl-CoA. Succinyl-CoA can you can hydrolyze. The thioester can be removed. It's basically a thioesterase reaction, right? And in so doing, via the succinyl-CoA synthetase, you can make some GTP. Whenever you see synthetase rather than synthase, it means that there's going to be a phosphate moving around. Here it's not ATP, but guanine diphosphate to guanine triphosphate. Why is that significant? It's because you want to keep a constant poise of purine and pyrimidine nucleotides for DNA replication and repair, and also for RNA synthesis. So this is going to be linked to all the other macromolecular reactions of the cell, as you might guess. Once you make succinate, then you can see this molecule looks identical, except that enzymes can determine this carbon from that carbon. These ratios here are just showing you how much of the carbon is lost per reaction. So you can radio label this pathway and determine where all the carbon goes. Okay, so succinate dehydrogenase doesn't have enough power, it doesn't have enough bioenergetic power because of the structure of this compound to make NADH, but it can make FADH2, or flavonoadenine dinucleotide reduced form. Once that happens, you've made fumarate, this double bonded compound, which will then pick up water and with the reaction of fumarase make L-malate. Malate has the right redox potential and oxidizing capacity to basically go through the dehydrogenase reaction to make oxaloacetic acid. So again, this gets oxidized, so NAD gets reduced. So malic acid is oxidized oxaloacetic acid, the ketone, and NAD is reduced to NADH. Now you've reformed OAA, you're ready to condense again with a single CoA and run the Tca cycle again. What you've done is primary net activity without considering all the other side reactions, which we'll build in later, is you've made 3 moles of NADH with this dehydrogenase here, the dehydrogenase here, the terminal dehydrogenase, the malate or the MDH here. You've also made FADH2, so you've made 4 different reducing nucleotide equivalents. You've also gotten rid of carbon dioxide, right? So you're starting to completely oxidize the carbon that you started with, completely send it out into the atmosphere, which will ultimately be recoupled to a bicarbonate reaction, an intermediate reaction, using biotin as a cofactor for the trapping of that CO2 and making bicarbonate, to then utilize the carbon back in the atmosphere. And plants do it much better, of course, than animal systems do or bacterial systems do, because they can photosynthesize using the energy from photolytic degradation of water to molecular oxygen. That's where we get our molecular oxygen from, except for the balancing acts. So that's basically the Tca cycle in a nutshell. So I'm going to stop there, right there, because we've gotten that far into the pathway. We set up all the players. And what we're going to do when we go to part 2 of this 4 or 5 part, not sure how far it's going to be extended. We'll figure it out when we get through maybe part 3 of this long mini subunit of these lectures on bioenergetics. Ultimately, we're going to be talking about several diseases, ultimately Alzheimer's disease, but along the way we'll talk about cardiovascular diseases, diseases associated with obesity like diabetes, and then we're also going to talk about cancers. But ultimately, we're going to land on Alzheimer's disease. But you need to have all this as background to know why ceramide, the sphingolipid, moving around ATP or sequestering ATP in the mitochondria can cause so many downstream effects. One of them can be the associated metabolism of proteins that are linked directly to late onset Alzheimer's disease, or AD. So to close this up, I'm going to stop here. We went 52 minutes, which is exactly what I wanted to do. That's my Facebook page, by the way. And again, you can catch that if you go to the very first slide. So all I've got to do right now is do my sign off, which is bye for now.